And we are live. Perfect. All right. I think we got the Instagram connected. We got the Facebook connected. Everything's connected. I think we're ready to go. Hello, everyone, and welcome to the inaugural edition of Rhythm of Research Live. My name is Gwyn McGuigan, and I'm the Manager of Digital Strategy and Content at the Canadian SADS Foundation. Thank you for joining us as we launch this new live stream series dedicated to bringing the latest clinical trial updates and research conversations directly to the inherited heart condition community. Rhythm of Research Live brings together leading clinicians, researchers, and industry experts to share the latest progress in clinical research and help bridge the gap between scientific discovery and the individuals and families these advancements are designed to serve. Our goal is to make emerging research more accessible by providing trusted, up-to-date information on clinical trials and the science that is shaping the future of care for inherited heart conditions. Today, we are pleased to welcome two distinguished guests. Joining us is Dr. John Giudicessi, a leading expert in inherited cardiac arrhythmias, cardiovascular genetics, precision medicine, and gene therapy. We are also joined by Dr. Whit Tingley, Chief Medical Officer at Tenaya Therapeutics. Dr. Tingley joined Tenaya in 2018 after serving as an attending cardiologist at the University of California, San Francisco, and holding leadership positions at Cytokinetics and Genentech. Together, they'll share updates on Tenaya's clinical research programs and provide valuable insight into the science behind genetics, clinical trials, and gene therapy. Today's discussion will begin with updates on Tenaya Therapeutics' ARVC, PKP2, and HCM, MYBPC3 clinical programs. We'll then explore the science behind these advancements, including the role of genetics in inherited heart conditions, how clinical trials are designed and conducted, and the foundation of gene therapy. Whether you're new to these topics or been following this research for years, we hope today's conversations provide valuable insights into where the field is today and where it may be headed in the future. Before we begin, just a few quick housekeeping reminders. If you're joining us live, feel free to submit your questions in the comments throughout today's discussion. We'll answer as many as we can during the Q&A at the end of the session. This live stream is being recorded and will be available afterward on our YouTube channel and Facebook, so you'll be able to revisit the discussion or share with others who may benefit. Finally, please remember that today's discussion is intended for general educational purposes only and is not a substitute for personal medical advice. We encourage you to keep your questions general rather than specific to your own or a family member's medical care. With that, it is my pleasure to welcome Dr. John Giudicessi and Dr. Whit Tingley.
Thank you.
Thank you. All right, there's a regulatory statement. So thank you to the Canadian SADS Foundation for Tenaya for allowing me the opportunity to present some of the highlights of our initial experiences with the TN201 and TN401 gene therapy programs. So I'm going to first present that data, and then Whit's going to come back and go over the genetics and the gene therapy basics to add some understanding. So we're going to start with the RIDGE trials, or the RIDGE studies, for PKP2 related arrhythmogenic right ventricular cardiomyopathy. This really contains two different studies. The first is a natural history and a seroprevalence study, helping to understand how many patients will be eligible. This is based on an adeno-associated viral vector, and there is some natural exposures. So this seroprevalence study has demonstrated that the majority of PKP2-related ARVC patients will qualify from that perspective. It's also gone on to show that a large number of clinically manifest PKP2-related ARVC patients continue to have a lot or high burden of PVCs and ventricular arrhythmias despite standard of care, which is what us as clinicians see is that this is a disease without disease-modifying therapy that has a high burden that impacts patients' lives. Then there's the interventional study. This is RIDGE-1. This is a phase 1b/2 gene therapy clinical trial. And we're going to go over data from the first 6 patients that was presented back at the American Society of Gene and Cell Therapy in Boston in April of 2026. And that has been shown that TN401, it's been well tolerated at both high and low doses. And patients undergo heart biopsies, both at baseline between week 8 and week 22, and then at week 52 at the conclusion of the first year of the study. And this is shown, RIDGE-1 has shown that TN401 at the RNA and DNA level it's reaching the heart, it's being expressed, it's producing the instruction necessary to make PKP2 protein. And I think most promising, although this was supposed to just assess safety, profiles, there is data showing that there may be early efficacy of this. And we're going to review that for the community because this is what is allowing Tenaya to continue to engage with the FDA and regulators on the design of a potential pivotal clinical trial and hopefully deliver what all those, both patients and providers, have been hoping for, which is a disease-modifying therapy for the most common cause of ARVC. Here's the overall design of the RIDGE-1 phase 1b/2 clinical trial. Again, the primary objective is safety and tolerability. So this study is designed just to assure that this does not cause harm. In addition, it's looking for dose finding to identify an optimal dose that's going to provide efficacy. And then some early effects or efficacy signals are assessed, and that's what we're going to review. The trial design involved 2 initial cohorts of 3 patients each. They're divided into a low-dose cohort, which is 3 times 10 to the 13th viral genomes per kilogram, and a high-dose cohort, which is 6 times 10 to the 13th viral genomes per kilogram. We'll review the data for the first 6 patients, but enrollment is ongoing in the expansion phase of the trial. And here's what the patient journey looks like. The first 8 to 12 weeks are screening. This is looking at things like the degree of RV dysfunction. How healthy is that right ventricle? Looking at the burden of extra beats from the bottom chambers or ventricular arrhythmias, and then making sure that patients don't have any issues with their kidneys or with the liver or with clotting disorders that would make them ineligible or make this therapy potentially dangerous. Then the patients that qualify, then entering the pre-dose and dosing phases of the study. Again, there's a biopsy that's taken before these patients receive a single one-time infusion of TN-401. Then 2 biopsies are taken during the follow-up phase, and these patients have multiple safety visits with us at the sites to be sure that we've got everything understood as far as from the safety outcomes. Then at the 1-year mark, they have a repeat biopsy and then enter into the long-term follow-up phase. Importantly, this fall, all 6 patients will be entering into this extended follow-up phase, and we'll have a lot more solidified data on the early efficacy of TN-401. Here's the initial demographics of the first 6 patients to receive the TN-401 AAV9 PKP2 gene replacement therapy. All were men, and this is important. PKP2-related ARVC is a male-predominant disorder, so this is not surprisingly. Their ages range between 27 and 56, with the youngest at diagnosis at the age of 16. All patients have implantable cardioverter defibrillators, and they have relatively severe arrhythmogenic phenotypes, with 4 patients requiring a catheter ablation as part of their standard of care therapy, and 4 patients having multiple episodes of severe ventricular arrhythmias, which are defined as sustained VT, which we'll discuss in a little bit here what that means, ventricular fibrillation, or unfortunately having that ICD go off appropriately. Also, all patients had a high burden of extra beats or PVCs from the bottom chambers, with a range between 618 all the way up to 8,634 PVCs per day, which is quite a high burden. And again, emphasizes that this is likely a more severe than the average PKP2-related ARVC patient. Here are where they are in April. 2 patients in the low-dose cohort had completed the week 52 activities. 2 patients, 1 in the low-dose cohort, 1 in the high-dose, were at week 32. And then the last 2 patients in cohort 2 were currently at their week 2 assessment. There were no and no ventricular arrhythmia. Of TN-401 related adverse events have been related to liver inflammation or elevated liver function tests or transaminitis. These have been mild, they've been in patients, and they've been manageable with increased dose or prolonged doses of steroids. At present, all patients have tapered off immunosuppressive medications and are back on their standard of care therapies. Now, this is what I'm talking about. So for patients in the audience or family members, you may hear the term PVC or non-sustained VT, and it's important to know what are we talking about. So premature ventricular contractions, if you look at the heart, here's what a normal heartbeat looks like. So you can see a normal heartbeat here, a normal heartbeat here, and this larger or more chaotic looking beat is a premature ventricular contraction coming from an irritable focus in either the left or the right ventricle, so those bottom chambers that serves as the heart's pump. When those PVCs string together, it's called non-sustained VT. As you can see here, 5 beats of non-sustained VT. Now, an increased PVC burden, but particularly increased non-sustained VT burden, clinically means that this is a patient that's going to be at increased risk for more rare but more severe ventricular arrhythmias, such as sustained VT, which is this VT then going up for more than 30 seconds. And if this is rapid, this often degenerates into ventricular fibrillation, which is a terminal rhythm. that results in a sudden cardiac arrest if this person is resuscitated, or tragically a sudden cardiac death if not. And so again, PVC burden, non-sustained VT burden, tells us that this is a patient that has an irritable electrical, or an increased risk of electrical instability that may be at risk for these potentially life-threatening events. That's then where we have the early data coming from TN-401. So this is looking at PVC burden. This is assessed by a 7-day continuous ECG monitor and then averaged and reported out in a per-day PVC rate or daily PVC rate. Screening or baseline is shown here on the left-hand side, and the patient's most recent here on the right-hand side of the screen. And as you can see, across all 6 patients, there has been a substantial reduction in PVC burden following dosing with TN-401. And this is an average of 62%. Now, I'll draw your attention to patient 5 and patient 6. These 2 patients in cohort 2, the high-dose cohort, had the highest PVC burdens of any patients in the study. They experienced a substantial and rapid reduction in PVC burden following dosing with TN-401 at their week 20 assessment. Now, I'm eagerly awaiting their week 32, their week 52. If this is sustainable, then this would suggest that there's potential dose-related effect, and this is really a significant reduction in PVC burden on top of standard of care therapy. And remember, again, these patients have defibrillators. They're on antiarrhythmic drugs. They've received catheter ablation, sometimes multiple catheter ablations. And this is on top of essentially throwing the kitchen sink at some of these patients. And so this is really quite a substantial and promising reduction. And we await that week 52 data. Here's looking at non-sustained VT. Again, I'll draw your attention to patient 2 and patient 5. These patients had a high burden of non-sustained VT per day across that 7-day continuous ECG monitor. Following the dosing of TN-401 or AAV PKP2 gene replacement therapy, both of those 2 patients had a substantial reduction in their PVC burden, which for patient 2 has been durable out to week 52. The remaining 4 patients had a low level of non-sustained VT at baseline that has been relatively stable pre and post dosing. And so then we'll turn our attention from PKP2 related ARVC to a different condition, which is myosin-binding protein C3 related hypertrophic cardiomyopathy. So instead of being an enlarged and arrhythmogenic right ventricle, hypertrophic cardiomyopathy is defined as a thickened heart wall muscle and a lot of symptoms due to increased pressure in the left ventricle. Again, 2 distinct studies, the MyClimb Natural History Study, which is actually looking at greater than 200 myosin-binding protein C3 related HCM patients who are under the age of 18, and really has demonstrated that there's a pretty significant burden of both symptoms, but also severe outcomes like sudden cardiac death, even in children who are myosin-binding protein C3 positive, and that is dose dependent. So those patients that have more than 1 copy that are defective, so 2 copies or 2 mutations in myosin-binding protein C3, really at a very increased risk of severe adverse events. And so this is just showing that in the pediatric population, there truly is a need for a disease-modifying therapy that can get ahead of this disease before it begins to progress and cause big, significant problems. MyPeak-1 is the phase 1b/2. This is the interventional trial. The data from the first 7 patients who received TN-201 was released in May of 2026. Again, like TN-401, it's been well tolerated at both high and low doses, and there's evidence at the RNA and DNA level that TN-201 is reaching the heart cells and providing the instructions necessary to make a new copy of the myosin-binding protein C3 protein. As far as early efficacy, all patients have had a reduction in LV hypertrophy as assessed by LV mass index, which we'll discuss here in a second. And subjectively, these patients, as assessed by New York Heart Association class and the Kansas City Cardiomyopathy Questionnaire, have seen subjective improvement in their symptoms. And with this data, Tenaya continues to engage with regulators in regards to the design of a pivotal trial to hopefully bring this towards patients into the market. Here are the overall demographics for the first 7 patients that received TN-201. This was more female predominant, so 6 females, 1 male, ages between 27 and 63. All patients had non-obstructive HCM at the time of their enrollment, which means that there's nothing blocking the blood flow from the left ventricle to the aorta. And these patients more struggle with the problem of increased pressure in the left ventricle due to what's called diastolic dysfunction. All 7 patients are symptomatic. all have substantial hypertrophy on their echocardiogram with their ultrasound of their heart, and all patients have ICDs. Now, again, encouraging safety data from MyPeak-1. It was well tolerated both the high and low dose cohorts. There's no dose-limiting findings, no evidence of myocarditis, and again, the adverse events were largely manageable and reversible, and largely related to increased liver function tests or transaminitis that was treatable with extended steroids and increased steroid tapers. All patients are now off immunosuppression, and over the course of both TN201 and TN401, we've learned a lot about the use of immunosuppressive regimens to reduce the side effects for AAV-related gene therapies that have resulted in less steroid use and thereby less side effects, and also good stewardship in the use of prednisone and steroids. Here's taking a look at the patient's subjective symptoms. This is first assessing New York Heart Association class. This is something that physicians use to communicate how's a patient's feeling to each other in just broader strokes. So class 1 symptoms mean this is a patient with no symptoms. They can run, they can jog, they can bike. They really have no limitation. Now, a patient with class 2 symptoms, these are mild symptoms. They are able to walk, they're able to go upstairs, they're able to do all activities of daily living, but they have to go run or they have to jog. They'll tell you, I just can't do that. I can't do it like my peers or others that I know of a similar age. And so that's just very relatively mild symptoms. Now, class 3, this is where patients are really beginning to be significantly limited. How I view this is that patients that will look at you and say, you know what, I avoid stairs. I have difficulty keeping up with my peers at work. I have difficulty keeping up with my spouse, and I avoid certain activities because it just makes me feel bad. That's New York Heart Association class 3 symptoms. Class 5, these are patients that are now unable to do a lot of physical activity. In fact, even walking down the hall to go to the bathroom can be a challenge, and they have symptoms at rest. Here's the New York Heart Association class in MyPeak-1. All patients began as New York Heart Association class 2 or greater. And at their most recent visit, 83% or 5 out of 6 have been reclassified to New York Heart Association class 1. Now, this is a subjective metric. We can begin to look at a more detailed assessment of symptoms using the Kansas City Cardiomyopathy Questionnaire. And I'll really have you focus on cohort 2. If you look at the cardiac myosin inhibitor trials, things like mavacamten, aficamten, for obstructive hypertrophic cardiomyopathy, we know that there was a placebo effect. So those receiving a sugar pill instead of the medication had a +6 change in their KCCQ score. If you look at all 4 or all 3 patients in cohort 2, you can see that there is an improvement over that placebo +6 in all 3 patients, suggesting that yes, indeed, there is an improvement from a subjective perspective in these patients' symptoms. Now, we don't just want to see subjective improvement. That's important. That's what patients are feeling. But we also want to see it correlate to some objective metric. And this is what this data is showing. Here's the data from MyPeak-1 looking at the left ventricular hypertrophy. This is measured as left ventricular mass index, which is just an echocardiographic or an imaging assessment of the total amount of heart muscle. To get an understanding of this, here's a normal heart wall muscle. As you can see, normal wall thickness in the septum, as well as in this wall over here. You can see this is a patient with hypertrophic cardiomyopathy. Very thick septum, also thickened at the apex, all around this heart. This is what's being assessed in that left ventricular mass index. And all 6 patients that have received TN201 have had a reduction in their left ventricular mass index over time. And for those patients now that are out in cohort 1, this has been a durable reduction in LV mass index. So here's a summary of some of the other parameters that are assessed. Biomarkers are important. Troponin, this is an enzyme the heart makes when it's damaged. Most myosin-binding protein C3 related hypertrophic cardiomyopathy patients have some degree of baseline troponin elevation. We can see that it's stable in 3 patients, but it's actually improved. It's gone down in an additional 3 patients. ProBNP, this is another protein that's produced when there's increased pressure in that heart wall muscle. It helps us assess for heart failure symptoms. And again, in 3 out of 6 patients, there's been an improvement. In 2 patients, it's actually gotten a little worse in that low dose cohort. But we can see, again, focusing on that high dose, most patients are either stable or improved. Taking a look at hypertrophy metrics, we discussed the LV mass index, improvement in all 6 patients, but there's other metrics like posterior wall thickness, looking at the thickness of that septum. And again, a large number of patients have had improvement in those hypertrophy indexes. We talked about the subjective New York Heart Association symptoms have improved. Kansas City questionnaire, again, for 4 out of 6 have improved, again, provides some early efficacy data that patients are feeling better after receiving TN-201. And lastly, some of this data is still pending, but looking at their walk times on a 6-minute walk, or looking at their peak VO2 on a cardiopulmonary exercise test, we're seeing some improvement in the 6-minute walk. We still await the CPET data for the patients in cohort 2, and I think that will be very promising, because if you're seeing the biomarkers improve, the hypertrophy improve, and the peak VO2 improve, then it's going to back up what those patients are saying, and saying there really truly may be a very clear early efficacy signal for TN-201, and that hopefully will be helpful as Tenaya, my colleagues at Tenaya, begin to have the discussions and continue discussions to move this towards a pivotal trial. And so with that, I'll turn it over to Whit, who's now going to go back and give you the genetics of ARVC, of HCM, and to discuss the kind of nuts and bolts of gene therapy and gene therapy trials. Turn it over to Whit.
Thank you, John. Great job. Very, very clear. And as you said, we're very excited about the emerging data from both of our gene therapy trials. Thank you very much to the Canadian SADS Foundation for this opportunity to present. And most importantly, thank you to everybody out there listening in the audience. This, of course, is for you. We really look forward to hearing your questions. We will take those at the end, so please, write them down. But it's important to us that you really understand as much as you can about your disease and your options. All right, so genetics and mutations in gene can lead to many health conditions, including heart disease and cardiomyopathy, or damage to the heart muscle in particular. Genes are instructions to make proteins. It's easy to think of protein as just a component of the diet, some sort of mixture of stuff, but proteins are exquisite little machines. And there are tens of thousands of them all working together in each cell to make each cell work, each organ work, and make us all live and thrive. A single letter change in the instructions in the DNA for a protein can cause that protein not to work and lead to disease. And in fact, that's the case in both of the conditions we're talking about today, hypertrophic cardiomyopathy and arrhythmogenic right ventricular cardiomyopathy. The most common genetic cause of ARVC is PKP2, and the most common genetic cause of HCM is MYBPC3. So that's part of the reason we've chosen these two particular genes to target for gene therapy. So ARVC is a condition that disrupts the normal electrical connection of heart cells, heart muscle cells, and it can also cause loss of the heart muscle cells and replacement with fibrotic tissue and fat tissue. This is a slow process that happens over time. One of the early manifestations of the disease can be arrhythmias coming from the ventricles as they scar. As John pointed out, these can start as single early beats, PVCs, and then get longer, non-sustained VTAC, ultimately VTAC, that can lead to sudden cardiac arrest through VFib. In the case of PKP2, mutations, genetic mutations, cause loss of function. So not enough protein is made, and so that function is lost inside the heart muscle cells. That leads to the disease process. What does PKP2 protein do? I think of it like a Velcro that holds the cardiomyocytes together. It's part of a structure of proteins called the desmosome that form at the attachment of heart muscle cells, and then they anchor the electrical connections between the cells called channels. Now, there are several genes that cause ARVC, and all of those work inside the same Velcro structure, the desmosome. PKP2, again, is the most common genetic cause. When these mutations happen, that Velcro is weak, it can pull apart, and the electrical connections, the channels can pull apart as well. So this leads to short circuits in the electrical connections, and that can lead to the early beats and the life-threatening ventricular arrhythmias. So the goal of therapy is to deliver a normal copy of the PKP2 gene to these exact heart muscle cells, allow those desmosomes to come together again, and restore normal electrical connections. Hypertrophic cardiomyopathy, John described the disease there. In this case, it's too much heart muscle. Some of these hearts sort of remind me of Arnold Schwarzenegger, just a huge chunk of muscle. But in the heart, that's not all good. In fact, it can make the heart too stiff. It doesn't fill well, and that limits the amount of blood the heart can pump. In some cases, that muscle gets in the way of the flow through the heart. That's called obstructive hypertrophic cardiomyopathy. But overall, the goal of therapy is to reduce that mass of heart muscle. And John described the direct measurements of mass we do through cardiac imaging and the encouraging results we have seen so far. MYBPC3 mutations are the most common genetic mutations leading to HCM. But there are other mutations, in this case, all affecting the protein structure called the sarcomere. The sarcomere is the combination of proteins that cause the muscles to contract. The main motor is called myosin, and here you see the myosin head in blue. You can think of it like a hand that pulls a rope. The rope is called actin. The more hands on the rope, the stronger that muscle contracts. And the target today, MYBPC, is responsible for determining how many hands are available to bind the rope. They can either let the head bind, as in the middle picture, or they can put the myosin head into what's called a super relaxed state, not binding, acting, not contributing to force generation. So the theme of genetic mutations that cause hypertrophic cardiomyopathy is too many hands on the rope. The muscle is just always pulling too strong. That leads those myocytes to get too big, hypertrophied. That leads the mass of the heart to go up, and that causes disease. For MYBPC3, the mutations prevent the production of C protein. And so on the right panel, you can see when C protein is missing, there's nothing to regulate the hands, too many hands on the rope overall. All right, so how do we fix these problems with gene therapy? So the gene therapies, in this case, are gene replacement therapies. So we have loss of a function of a gene. So we want to add in a full-length wild-type copy of the human gene to restore the normal gene function and restore the normal protein function. So this is targeting the underlying or root cause of the disease directly at its source. And the goal is to stop the progression of disease and in the best case is actually reverse disease. So improving heart rhythm, improving the heart's blood pumping output, improving symptoms, of course, exercise capacity and quality of life. Now these gene therapies only need to be given once. So a single intravenous infusion can last, we've demonstrated, 2 years. Other data from other gene therapies for the heart have gone out 3 to 4 years. And the initial gene therapy trials have shown efficacy that can be durable even many more years than that. So we need more time, more follow-up from those dedicated first patients in our trials. But because of the way the heart works, we do expect and hope this could last for decades. We get this question a lot, so it's important to say this gene therapy is treating the heart, but it does not treat sperm or eggs. So parents who are patients still pass on the genetic mutation to their children, and their children will need to be treated separately. All right, how does gene therapy work? As John mentioned, we take advantage of something that occurs in nature, a virus called an adeno-associated virus. A key advantage of this virus is it doesn't cause disease itself. We have many, 15, 20% of adults have been exposed to this adeno-associated virus, but it doesn't cause disease or symptoms. The other advantage is that it's nature has designed it to deliver DNA to cells, including heart muscle cells. Very effective at that. So that's the capacity we want to retain. There are different versions of AAVs. We use AAV9, which is a naturally occurring type that has a natural ability to get DNA to heart cells, and is also the most widely used vector for gene therapy, and there are approved drugs using AAV9. So how do we make the gene therapy? Well, from AAV9, really all we want is the capsid, the package. It's like a delivery box, delivering what you want in the mail. So we take out all the viral contents of the box, and we put in what your heart muscle cells need, the DNA for PKP2 or MYBPC3, along with a promoter to turn it on in the heart muscle cells. We package this up, and then we deliver it. As I said, it's a single intravenous infusion. So again, we're using the delivery box, the viral capsid with the DNA that you need inside it. We give it intravenously. It goes throughout the body, and it has a natural proclivity to go to heart muscle cells, shown here on the right. The cells take up the package, bring it into the nucleus where DNA is utilized. It takes the DNA out, it destroys the package, so the viral pieces get destroyed, and the DNA is left, and that is used to produce the protein. The process of protein production is called translation. So the DNA is translated into protein. There is a key intermediate step called RNA. So the DNA is transcribed into RNA or messenger RNA, which then makes protein. And we highlight the RNA here because it's something we can very specifically measure in the heart to see if the gene therapy is doing what we intend it to do. So here we are looking at the RNA levels from heart muscle biopsies that we've taken from patients after they've been treated in the RIDGE-1 trial. So you can see on the left, RNA is clearly detectable in all patients tested to date in both the low and the high dose cohort. The numbers here, like 1.8 or 5.0, indicate how many, I'm sorry, on the left here, we're talking about the DNA. So on the left, how many copies of DNA are present per nucleus or per cell on average. And so a number of 5 means we're getting about 5 copies per nucleus in the heart. These numbers are consistently higher than what were needed for max efficacy in the animal studies we did before the clinical trial. So very high levels of cardiac transduction or DNA delivery. On the right is the RNA that I was talking about before, and you can see across all patients tested to date, we can see RNA being expressed. This is RNA specifically coming from the therapeutic TN-401, and it is demonstrating that it is indeed getting into the nucleus, and the DNA is activated and turning into RNA, and it's the RNA that's used to make protein. The measurement of protein is more complicated because all patients have protein at baseline, and because we're making a fully normal protein, it's indistinguishable from the protein patients already have. But the RNA data is particularly encouraging that we are delivering the gene and getting the expression as designed. This slide is showing similar data for TN-201, in particular focusing on the messenger RNA production in the hearts of the patients that have been treated with TN-201 for hypertrophic cardiomyopathy. And again, we can see that messenger RNA is being made across all patients tested to date. So successful transduction and expression of the MYBPC3 gene. Now, there are potential risks to AAV-based gene therapies, and these are certainly important to know. First and foremost, the immune system can't tell that this is the therapy. It assumes it's a virus and tries to get rid of it. This causes inflammation, and the inflammation itself can cause damage if not controlled. We use immunosuppressive medications to reduce the immune response. We start them before the treatment with the gene therapy, and then we gradually taper them off after the gene therapy. Both TN201 and 401 can lead to inflammation in the liver in particular, just like other AAV gene therapies. This is well understood in the field. It's also understood how to prevent and manage it. And steroids, such as prednisone, are the key drug to use. We also use a drug called sirolimus to help limit the amount of steroids that we need. Other types of serious inflammation have been seen with other AAV-based gene therapies and require very close monitoring. So all patients in our trials get frequent blood tests to make sure these other types of inflammation aren't occurring, and we have therapies we can use if needed. In some cases of gene therapy, the immune reaction can prevent the treatment from working as intended if it blocks the gene therapy or removes the cells that are using the gene therapy. So that's another important role of the immunosuppression that we use. Important thing to know is that at this time, you can only get an AAV gene therapy once. Once your immune system has seen it, it develops very effective ways to block it using antibodies. And therefore, it's a one-time dose, but one time only. Various scientists are working to find ways to administer second and third doses, and we look forward to more progress in that area. AAV gene therapy can deliver a working gene to the incorrect cells, so that AAV9 capsid I mentioned is very good delivering to heart muscle cells, but it also delivers to the liver and other cells. So we need to do a special trick to make sure the gene therapy only turns on in heart muscle cells. That trick is called a promoter. I'll get back to that. Of course, these gene therapies are still under investigation. They are not approved by any health authority around the world, and we do not know all the potential risks to date. We do at Tenaya take a very comprehensive approach to safety. So, for example, the gene that we use is a full normal copy of the human gene. That's important because it means that the immune system will recognize that it's normal and not react to the therapeutic protein, so to the PKP2 or MYBPC3 that we're adding. And we design these to work only in heart muscle cells. We have used a promoter which only turns on that gene inside the heart muscle, not other types of muscle cells and not the liver. As I mentioned, we use the AAV9 capsid. This has been the most widely used capsid for gene therapies, with over 5,000 patients treated around the world. The dose is important as well. We are fortunate that we are seeing transduction expression and efficacy at doses of AAV lower than other gene therapies, including approved gene therapies that have treated thousands of patients. The larger the dose, the larger the immune reaction and the more immune suppression that you need. So the fact that we're using lower doses, as John mentioned, in the E13 range, so 3 to 6 E13 vector genomes per kilogram of body weight, those relatively low doses reduce the amount of immune suppression that we need and reduce the risk of those more serious immune reactions. Important to say, again, that the immunosuppression is temporary and that we tailor it for each patient so that no patient is getting more than they need to control their own immune system. To do this, we do very frequent monitoring, weekly blood tests at the beginning. As we reduce the immunosuppression, we follow the liver in particular very carefully. If there's any signs of inflammation in the liver, we keep going a little bit longer with the immunosuppression. But this is the way we can tailor it for each patient, make sure each patient is safe, but also not use more immunosuppression than we need. If you are considering participating in a gene therapy trial, here's some food for thought or things you can learn. First, will it fit into your life? There are frequent visits and frequent blood tests at the beginning, as I mentioned, all patients are required to complete those assessments. What expectations do you have about the outcome? This is an unproven therapy at this point. These trials are designed to show the safety and efficacy, but that is unknown at this time. So the results may not turn out as you're hoping. How will you feel about that if that is the case? Certainly very important to understand the risk. Make sure you spend a lot of time with the study doctor getting your questions answered and reviewing the various materials. You really want to know before getting into this what the potential risks are, and then weighing the potential benefits and risks before making your final decision. Important to have high confidence and trust in the clinical team. We have chosen to work with expert centers around the United States and the United Kingdom, including, of course, the Mayo Clinic meeting, a leading medical center for both hypertrophic cardiomyopathy and ARVC. And then who can help you with your decision-making? Your primary care team, for example, your family support network. Certainly involve others and get their perspectives as you're thinking about participating in a trial. If the trial is not for you, there's certainly other ways you can participate and help the process of developing new and innovative drugs for your condition, hypertrophic cardiomyopathy or ARVC. And I'll say patient advocacy, like the Canadian SADS Foundation, is fundamental. The ability of patient groups to influence us drug developers, the academic medical centers that do the trials, and the regulators such as the FDA and Health Canada that make decisions about which drugs to be developed, how they're developed, and whether or not they're approved for clinical use. We all listen to patients. Patients can help by relaying their experience, and even more valuable, participating in natural history studies like the RIDGE and MyClimb studies that John just mentioned. The more we understand about the course of the disease, the more we know what you need next to help you thrive despite your disease. In addition to being a participant in a trial, we rely on patients to review the protocols, help us decide, where we might make it less burdensome for patients, for example. Also to review the informed consent form. Are the risks and potential benefits clearly described? And educating FDA, Health Canada about what therapy means to patients. Ultimately, in approved drugs, patients and patient groups can help reduce barriers so that these drugs can get to all patients who need them. So thank you to everybody in the audience already participating in these types of activities. Again, really fundamental, really helps move drug development along and get new treatments to patients like you who need them. So resources you can reach out to here, of course, the Canadian SADS Foundation, first and foremost, and their website is listed here. Dr. Giudicessi and his team at the Mayo Clinic are, again, experts in both of these diseases. We have an address here that you can contact, in particular about participating in these types of activities. And please feel free to reach out to us here at Tenaya, patient.advocacy@tenayathera.com. We have a council of patients, again, reviewing our documents like protocols and consent forms and giving advice about how best to develop our drugs so that we can address patients' biggest needs. We also do on our website have some brochures and substantial amounts of information about HCM and ARVC. So with that, we can get to the most important part, which is answering your questions. So please submit them now.
And I will also bring myself in. For those who are watching, if you need a second, I figured I could maybe start with a couple questions. So first of all, I want to say thank you so much, Dr. Giudicessi and Dr. Tingley, for sharing your expertise and insights with our community today. I guess one question that I know our community is very interested in learning is for someone living with ARVC or HCM, there's always the question of what does this mean for the future? I was just wondering if one of you could kind of go into detail about how these types of research programs bring us closer to potential new treatment options.
Yeah, no, I mean, I think this is what the communities have been waiting for, right? And so I've been very fortunate. I'm more towards the beginning of my career. I've been practicing when these have begun to percolate and make their way through. But you go back and you talk to some of my more senior colleagues who've been practicing for 20 or 30 years, and they'll tell you, they're just putting out fires, right? They're waiting for a bad arrhythmia to happen. They're waiting for a heart failure event. And finally, these trials, I think, are giving us the first hope that maybe we can do something prophylactic, something preventative to actually prevent that. And I think that's reflected in who participates. I think, you look at patients who choose to participate in these trials, and a lot of times there's not an expectation for them to gain benefit. What they really want is that they want this to move forward for that next generation. And I think that's a really good motivation, because that's what all of us want. We want a brighter tomorrow. We want the patients we're taking care of today. We want their children and their grandchildren to have a different course, right? To not have gone through all the ups and downs that those patients have experienced in their journey. And that, at the end of the day, is, I think, what we're all hoping for, that this is the beginning, right, of that sort of trajectory.
No, I couldn't agree more. I know I've heard before the quote that participation in trial today is the gift to the future tomorrow. So I couldn't agree more. I think that's a fantastic statement. I have another question here. I know genetics can be very, very complicated. Would either Dr. Giudicessi or Dr. Tingley be able to explain more about how knowing the specific gene variant helps doctors and researchers better understand their condition? And then with that, hear a little bit more about how sometimes people may have the same genetic condition, but have different experiences with their diseases.
I'll let Whit start, and then I'll chime in.
Yeah, no, you're quite right. Genetics is complicated, and the more you learn, the more interesting it gets. And many times it does come down to exactly which letter is changed or which deletion that you have. A key theme to know, if you're interested in genetics and how it affects gene therapies, is the concept of loss of function versus gain of function. So a misspelling that causes loss of function for it typically means there's just not enough protein made, or the protein that is made is unstable and it gets broken down. So the cell doesn't have enough protein. And so the theme is replace. And so the gene therapies we talked about today are replacement therapies. Just give a new copy of the gene and restore the protein. But other mutations can cause gain of function, and that can mean a gain of abnormal function or dysfunction. And so adding more won't do anything if you have a poison peptide, a protein that's just disrupting the cell. You got to remove that protein. And that takes different types of gene therapy. We are very excited about the potential for gene editing technologies like CRISPR, people may have heard of. There are a few more barriers to getting that into the heart and working. Certainly in animal models, it works very well. So we look forward to the field being able to address that type of mutation as well, but it is further away. So in both of our genes, MYBPC3 and PKP2, the vast majority of disease-causing mutations are loss of function mutations and will respond to the gene replacement therapy that we described.
And I'll just echo, I think the cardiomyopathies, one of the bright spots is that a lot of the genetic cardiomyopathies do fall into this loss of function category, right? They're often caused by what we call truncating variants, right? So there's different, right, missense variants, there's spelling errors. You can produce a functional protein, but it doesn't work how it's supposed to, right? But truncating variants, frame shift variants, nonsense variants, splice site variants, they simply reduce the amount of functional protein. And that's where these gene replacement therapies, really come into play, right? And then Wick got into the more gain of function. You can even have loss of function poison peptide, which we can see for myosin binding protein C3, where some patients do have missense variants that can do that. Those are more challenging, right? And so that's the next phase, right? The next generation of gene therapies to really get at ways to get around that problem, right? And there's gene silencing, there's gene editing, there's way to suppress and then overexpress. There's all these different mechanisms. So what I'll say is the most important thing it comes down to is genetic testing, right? You've got to get the genetic test, because we're entering an era where not only are you have clinical trials in phase 1, but look, you may have trials that are entering pivotal, right? And you do, right, out there for Danon disease, right, which is a form of, you can think of hypertrophic cardiomyopathy. there are trials out there. So I think the important thing to take away for patients is if you've got a diagnosis of HCM, if you've got a diagnosis of arrhythmogenic cardiomyopathy, and you haven't had genetic testing, it's really, really important that you get that because it may open doors, right? And it may not just open doors to participating in a clinical trial, but how us as clinicians manage you. A PKP2, arrhythmogenic right ventricular cardiomyopathy, is very different than a lamin-related cardiomyopathy or a filament C-related cardiomyopathy. And then same thing for hypertrophic cardiomyopathy, PKRG2 syndrome, Danon disease, very different than myosin binding protein C3-related HCM. So right now, conventionally, it'll change how we manage you. But even more importantly, in the future, the hope is that we're going to molecular diagnoses and we're going to molecular therapeutics. And that's why the genetic test and what variant is in there becomes so important.
And staying on the topic of gene therapy, I don't know if one of you'd be able to kind of explain more about like kind of the biggest challenges that researchers such as yourselves are still need to overcome before these treatments become more widely available.
Yeah, so gene therapies like the ones that we've talked about have been approved, for example, for spinal muscular atrophy or Duchenne's muscular dystrophy. So these can work, and the hurdles have been removed. For the two that we talked about today, or more generally for cardiomyopathy gene therapies, we just need to do the work to prove that we can give them safely and that they're doing what they're supposed to do. We start slowly, one patient at a time. We've moved out of that phase in our trials. Now we can dose multiple patients at a time, but still, relatively small groups of patients that we follow very closely and do carefully. Our next step will be pivotal trials. And so that'll be another group of patients, a somewhat larger number of patients. And those are designed to have treated enough patients that at the end, we know the drugs work and we know how to give them safely. And that's when they can be used clinically, approved by Health Canada and the FDA and used clinically. As we go forward and in the last, a couple of years of AAV gene therapies, we're not seeing new unexpected safety events. So we actually pretty much know what to look for now. We talked about inflammation of the liver. We talked about other types of inflammation, complement activation being one of those. We actually know what to monitor and we know how to prevent and treat it. We're getting better and better. So with each new gene therapy, it's just a little bit easier. We kind of know how to start and we're not having many surprises, which is very encouraging.
Oh, fantastic. And we may have talked about it already, but I know one thing that I wanted to kind of go over once more. I love to always get in as layman terms as possible. But for those who have been diagnosed with a genetic condition like PKP2 or MYBPC3 associated HCM, does that mean that any person that has been diagnosed may be eligible for these trials, or what other factors do researchers consider when they're speaking with patients that show an interest in participating in trials?
Yeah, I think that's a great question, right? So you're really looking at a sweet spot, right? For these gene therapy trials where you can't have what we call gene positive, but phenotype negative, right? So you just have the gene, but you don't have the disease. You're not going to qualify for those trials because we have to have some disease to show we have improvement, right? So that's a swath of patients, especially for PKP2. That's a large amount of patients that just have the gene but don't have any real clinical manifestations. So those patients are removed. Other end of the coin, you have patients where their disease is advanced, right? This is more end-stage disease, very advanced. If you look at PKP2 ARVC, another good example, if that right ventricle is very enlarged, very dilated, there may not be enough heart cells, living heart cells left to transduce with the gene therapy for us to get a viable outcome. So you're looking at that patient in that sweet spot where they've got disease, right? But it's not too far that we think we can bring it back or we think we can help, right? So that's the first thing. That's what all those enrollment criteria, all those check boxes that us as site investigators, as the sponsors, we've got to go through. That's what that's trying to align to get that this is an ideal patient. But then you have more personal things. So one of the things that I sit down and I talk about to patients is, look, these are very intensive, right? For the phase ones, we've had to put patients in the hospital between 7 to 10 days, right? That takes somebody that has a fair bit of time, right? That doesn't have a job that requires them on site, 24/7 kind of deal. In addition, the follow-up, especially for the first 3 to 4 months, is very, very close, right? So it's a follow-up visits essentially every 2 weeks there for, a couple months. So you've got to have somebody that, has the flexibility in their schedule and has the willingness, not just of them, but an employer for these younger patients to allow them to attend those visits and give them the time off. So that's something you've got to consider as well. And then I also think about how close are you, right? Because look, these sites are all at major quaternary tertiary medical centers. Yes, there's likely one that's near you, but still, I don't want a patient traveling 8 or 9 hours to get to participate. So we have to think about that too, right? The practicality of it. If you're so far remote from one of these sites, is it actually going to become a burden? You might think up front, yep, it's fine. But as you get into the grind of the 2 or 3 months, is this going to begin to wear on you? So you don't want it to really impact that person's quality of life. And so you've got to put those factors in too. So there's a lot of the soft side of stuff that we have to consider when we're looking for what is the ideal patient to put in. And I always go through patients, I tell them, you need to think about this too, the impact of this, before and know what you're getting into. And this is what Whit went over, before you sign on the line to participate in these trials, because it does require a lot of lifting for patients. And we want them to know what they're getting into. So we have a really frank conversation before we'll put anybody in one of these trials about what are you getting into.
Yeah, and the patients in our trials are heroes. No other way to put it. They are sacrificing an amount of time. They're taking on an amount of risk, especially in the early phases where there's less that we know. We are glad they're feeling better, as John said. So it's been helpful for them, but it's also helpful for patients today and patients of the future, as you pointed out. So definitely heroic. We do think these factors will change and get easier as we go forward, and more patients will be eligible. So as we've described, we monitor the immune system very closely, and we start to see patterns. And then we can figure out, well, we really only need the blood test on this day or that day, or nothing happened in the hospital. We don't really need to hospitalize patients anymore. These are the kind of things we hope to see, so that when we get to the next trial, the pivotal trial, the burden will be reduced substantially, but the safety will be maintained. And then in terms of eligibility, John described the sweet spot. Patients that have a high disease burden now and that have the most to potentially gain from a gene therapy, ultimately, it would be well suited for a preventative therapy, one IV dose that then lasts for years. In the future, the best time to get the gene therapy would be the day before you get the disease. And it's important to know then who is going to get the disease from those who may never get the disease. And that's part of the reason that we are so thankful for the patients participating in the natural history study. The more we learn about what predicts who's going to have bad disease, the more we can identify those patients that will benefit most from preventative gene therapy.
No, that's fantastic. Thank you so much. Well, it looks like we're coming just about to the end. So I wanted to give one last thank you so much, Dr. Giudicessi and Dr. Tingley, for joining us today and for sharing your expertise and insights. We really appreciate the time you took to discuss Tenaya's research programs and to help our audience better understand the science behind genetics, clinical trials, and gene therapy. Just a reminder to everyone that's joining us today that this live stream will be available afterward on our YouTube channel and our Facebook, and will remain available as a resource for you to revisit, share with others, and continue learning about the latest developments in inherited heart condition research. Thank you again for joining us for the first edition of Rhythm of Research Live. We look forward to bringing you more conversations with experts from across the research community in the future. Thank you so much, and we'll see everybody soon.
Thank
Bye
you.
now.